Hello, everybody. Good morning. So today I'll be talking about why the aging brain is so interesting and so mysterious, and how a social and affective neuroscience approach has started to begin to solve one of these mysteries. So if you don't know much about aging, it's actually awesome. So getting older is great in a lot of ways. So as people get older, they experience more positive, less negative emotion, report better controlling their emotions, and actually in many cases do better regulate emotions. But getting older is also tough. So there are declines with age in fluid cognitive abilities, things like working memory, source memory, inhibiting interference, cognitive flexibility and control. These things decline quite linearly with age, starting actually in the late 20s throughout adulthood. And these declines have been linked to changes, gradual changes, structurally and functionally in the prefrontal cortex. So these fluid cognitive declines have been well characterized neurobiologically. But the improvements in emotional experience, in well-being, have not been characterized neurobiologically. So what? The behavior is pretty interesting by itself. But what this creates is a sort of bizarre dualism, where you actually read papers in psychology and aging that in the discussion say, well, it's unclear whether these effects are due to biological declines with age or motivation, as if motivation's like in the ether or something. It's unclear where it is or what it is. If it's not a brain change, what is it? So this is just a little weird. I started reading stuff like that early on in graduate school, I'd say in my second year. And I talked to my advisor, who was Laura Carsonson at the time, and I was like, this is strange that people are like being so dualist. We should like write a review about this and try to figure out like, well, what are the brain systems underlying these changes in motivation and emotional experience with age? And she said, based on what data? So I think you have like a decade of work to do to try to figure this out. So here I am 13 years later with like a little bit of data to try to start to resolve some of this. So we're making some progress, but it takes a really long time. So I started out studying decision-making in aging. That's mostly what we study in the lab. And so studying decision-making, I'm studying both these affective functions and things like emotional control, but also these fluid cognitive abilities are also really important for decision-making. And the early research on decision-making in aging showed both improvements in decision-making and declines. So one example is in intertemporal choice tasks, older adults are more likely to take a delayed option, so take a larger reward at a delay than younger adults, so arguably making better decisions. But in learning-based tasks, older adults are slower to learn. So in reinforcement learning tasks, older adults learn more slowly, actually make more mistakes. And those changes in reinforcement learning have actually been directly linked to changes in the dopamine system. So older adults have lower levels of dopamine, fewer dopamine receptors, and actually in pharmacological manipulations with dopamine precursors can actually recover reinforcement learning in older adults. So those changes in learning seem to be associated with declines in the dopamine system, but what about those improvements in decision making? People have also associated those with declines in the dopamine system. So one hypothesis is that older adults don't have much dopamine, so they don't have much motivation. So the reason older adults take delayed options in an intertemporal choice task is because they're just not excited about the reward that's available right now. So you just can't get old people excited about stuff, is the idea, because there's not dopamine there. I think you probably can already tell that that just didn't seem right to me. So the question here is it just harder to get excited when you're old? And no, it's not. So first bit of behavioral data I'm going to show you shows flexibility in intertemporal choice. So here we compared 3 different intertemporal choice tasks, so making a decision between a smaller, sooner reward and a larger, later reward in 3 different domains: a financial domain, a social reward, so you identify a social partner that you wish you spent more time with, and the magnitude of the reward is how many minutes you're going to spend with them. A little bit of time now, more time if you wait. And then there was a hypothetical wonder drug that improves cognition and general organ function, and it was a smaller dose now, a larger dose later. This is all hypothetical. But what you see is that, and on the y-axis there is the proportion of sooner options. So here, older adults are actually more impulsive, more impatient. They're taking the sooner option, they're discounting more. So there are behavioral findings in the monetary domain with money or points where older adults discount less, so that plot on your left usually is sometimes a declining line. It's basically flat in our data. And what you see is actually older adults want these social and health rewards now. So they do get excited, but about different stuff. So different kinds of rewards can actually make older adults more impulsive, more impatient in intertemporal choice tasks. So first, just flexibility in this task. So what's going on neurobiologically? So I mentioned before that behavior is really interesting, but there's no sort of brain to it. And so if we look at maybe older adults are using different systems or not using the same systems quite as well as younger adults, in the same domain in intertemporal choice tasks, but also in other related discounting tasks like probability discounting, physical effort-based discounting. So across 3 different decision-making tasks, we looked at representations of value, representations of subjective value in adults between the age, from the young 20s until the early 80s. We see a remarkable lack of age differences. You can actually lower the threshold quite a bit and not see any age differences across these tasks. Instead, what you see is that across adulthood, adults of all ages are representing subjective value strongly in the medial prefrontal cortex. And you can actually break it down into smaller age groups, and you see basically the same pattern of activity. So evidence for functional preservation of these systems. But it's also possible that we see activation in these systems, but they're accounted for by different neurochemical systems. Maybe there actually is less of a dopamine influence here, but I can't see it with fMRI. And so as part of a series of studies that we were beginning using PET imaging to look at dopamine system across adulthood, we wanted to do, actually, honestly, the reason we did this is because we just needed to do a sample size calculation for a grant. So instead, we decided to do a meta-analysis to get a meta-analytic estimate of the age effect on decline in the dopamine system. And so we ended up, this turned into a larger project, obviously. We ended up looking at, so D1 and D2 are different receptor subtypes in the dopamine system. DAT is a dopamine transporter. And so just focusing on those, on the y-axis there is the correlation with age. Strong negative correlation. So across over 75 studies for those, actually over 80 studies for the receptors and transporters, you see strong declines with age. So lower levels of dopamine receptors of both types, lower levels of transporters, but preservation in dopamine synthesis capacity. So this diamond that you see that where the confidence interval crosses over 0 shows that's no effective age. So at least no evidence for an effective age. So basically, one possible way that the dopamine system could be working well is with preserved synthesis capacity. So presynaptically, there's dopamine being produced and potentially released. But the problem is, if you look at these receptors here, there are fewer postsynaptic sites for that dopamine to act, so a barrier. So when we first saw this, it was really interesting, because I said, oh, well, maybe with preserved synthesis capacity, dopamine can work well in certain contexts, but there's this postsynaptic barrier. So what we decided to do is break this down, look within a receptor subtype and see, well, are there parts of the brain where this preserved synthesis capacity does have potential for action? So are there parts of the brain where there are preserved receptors so that we could actually have a somewhat intact pathway in old age? I should also say this was done by Teresa Carrer, who just sent me a random email and said, oh, I have some funding to do a master's thesis wherever I want. Could I come and do that in your lab? And I'm usually horrible at responding to those emails. But I responded to that one. She came and did this meta-analysis in 1 year and published it. And she actually just published a serotonin meta-analysis like in the last few days. So respond to those emails. They can be amazing. Okay, and so this looking at regional variation in D2 receptor availability was led by Kendra Seaman, who's a postdoc in the lab, is headed soon to a faculty position. And what she did was really break the brain down into a bunch of different subregions, which people hadn't really done before with PET imaging. And so this shows you percent differences per decade. So this is actually pretty hard to read. But I'm going to zoom in on a couple sections of it here. The summary is that across 2 data sets, so we combined 2 data sets, and we found the largest age differences, these negative age differences, these negative declines that we see across the brain. These largest effects were in lateral frontal cortex and other lateral cortical regions, and the weakest or no effects actually in ventral striatum, pallidum, and in some medial frontal regions as well. So just to show a couple of those, at the top you see postcentral gyrus, middle frontal gyrus, superior frontal gyrus, with declines of 12 to 16% per decade estimated from this cross-sectional data. Whereas, and there's a vertical dotted line here, which is an age effect of 0. And so a couple of regions are not significantly different from 0, like pallidum, hippocampus, ventral striatum, but also subcallosal area, pregenual frontal cortex also shows really shallow decline with age. So this frontostriatal ventromedial circuit seems to be somewhat intact, or at least there's preserved levels of receptors there. So given what we know about intact synthesis capacity, potentially this is a subcircuit where dopamine can actually act quite well even in older age. This is just a little bit of a showing off thing here. So Kendra also created a web app, so you could analyze all of our data by regions if you're curious about any of these things. You can look at linear quadratic effects. So you can just scroll through this. And so there's the link if you want to check this out and mess around with our data. You can also see the output of the regressions. So this is really fun. Thanks, Kendra, for making that. OK, so the question I sort of started with before, or this proposed theory was, is there global motivational decline with age? No, there's not, right? At least the behavior shows that quite strongly, and it had shown that for 20 years before anyone was doing neuroeconomics of aging. But what we started to do is identify functionally intact circuits and actually dopamine-mediated functionally intact circuits with age that seem to be especially functional when you're using rewards that are goal relevant for older adults. So there's evidence actually for motivational function across these circuits. So given that evidence, we're starting to do some other studies to try to see if, taking what we've learned about these reward types and these brain circuits, can we get things like reinforcement learning or even other cognitive tasks to work better using these type of rewards? And so we have studies comparing social and monetary rewards. So for example, just looking at basic responsiveness to these different kinds of rewards, but also in reinforcement learning tasks using these kinds of rewards as feedback. And so here's an example of some stimuli that we use where you can make some money or lose some money, or you can do something that makes this old guy really happy with what you'd ever just did, or makes him really mad at you. And so we're using these dynamic stimuli as rewards, as socio-emotional rewards in these tasks to see if they better motivate learning and other cognitive function with age. These dynamic stimuli are a public resource available to all of you. So they're these dynamic faces database. And so they're freely available. They're hosted in the library of the Max Planck Institute. But you can find them. This project was actually led by an undergraduate in my lab who created all these stimuli from actually high-resolution stills. But these are all available. One caveat, they're all white German people. So that's kind of a big limitation of the data set. But the stimuli have great norms, and they're really nice. OK, so that's about all I have to tell you today. I just have to give a shout out to National Institute on Aging, which would be impossible for me to do any of this very expensive PET imaging work without their support. And thank you to my lab. Thanks, everybody.